As a kid, I wanted to be a hunter. Plants are very fascinating organisms, very unique.
I must say that in the first meeting that we had, it was like 1 month after we
Yes.
arrived, we were very lost.
Well, I am very happy that you have joined us in EpiSeedLink.
Very important thing is you need to fall in love with your project.
A scientific career is a marathon, but there are sometimes very special or scary moments. Hello and welcome to EpiSeedLink's podcast series. I am Nora and I am a PhD fellow and I am currently working at the University of Galway.
Hello, my name is Mayra, and like Nora, I'm also a EpiSeedLink fellow. I'm working at the University of Amsterdam. Today we are here for one more episode of our EpiSeedLink podcast.
We are very happy today to interview Dr. Aleš Becinka, part of our EpiSeedLink consortium. Aleš is a plant geneticist with a long and fruitful career under his belt. He obtained his PhD in 2005 at the Leibniz Institute of Plant Genetics and Crop Plant Research in Germany. Thereafter, Aleš stayed at the Gregor Mendel Institute of Molecular Plant Biology in Vienna, Austria, as a postdoctoral researcher, followed by 7 years as the group leader for plant breeding and genetics at the Max Planck Institute for Plant Breeding Research in Cologne, Germany.
Then he returned to home, to the Czech Republic in 2017, and is currently a group leader and deputy head at the Center of Plant Structural and Functional Genomics at the Institute of Experimental Botany in Olomouc, Czech Republic. I hope I told the name of the city correctly.
Perfect, it's Olomouc.
Okay. Olomouc. Okay. His group is focused on plant chromatin organization and function. He has over 75 journals, publications, and 4 book chapters. It is not a surprise that we feel very honored to receive Aleš in this podcast for EpiSeedLink. We are very lucky to have him be a part of the project's external advisory board, giving very valuable insight to the fellows. Welcome, Aleš. Thank you for being here.
Welcome, Aleš.
So thank you very much, Nora. Thank you very much, Mayra. It's really honor for me as well to be part of the EpiSeedLink podcast, and I'm really grateful to be here today and to have a chance to be discussing and chatting with you.
Okay, the first part of our interview cannot be anything different. First of all, could you give briefly us some details about the main research project you are developing in your lab?
So my team is focusing on chromatin organization and function. So we are interested in how DNA is packed in the nucleus, how it's assembled into chromosomes, and what proteins interact with the DNA, and how they help to regulate transcription, how they put it together, and also how basically DNA repairs itself using the proteins which are encoded in its sequence.
During the beginning of our career as PhDs or postdocs, we usually navigate through different topics and question ourselves on which topic we want to focus on. We are curious to ask you about how and when did you come to research plant chromatin organization and function? And if you could tell us a little bit about your own research story.
So my research story is a bit complex, but also interesting, I would say. So my initial intention was not to study plant chromatin. As a kid, I wanted to be a hunter, and later, because my great grandfather, he was always interested in nature, and he took me, to nature and to meet his friends, which were hunters, and I was impressed by those guys. And later I wanted to be zoologist, but both hunters and zoologists, they often kill animals. So that's what I did not want to do, and I felt like working with plants is maybe nice because I don't need to hurt them so much, and that's why I studied botany. So at the university I did systematic botany studies, so kind of like plant taxonomy. And afterwards I found out that the taxonomy, at least at that time, was very descriptive. It was happening at the nice locations, I could go to nature, but at the end of the day I had feeling that I'm understanding very little of what is happening in the plants. So that's why I went for my PhD to IPK Gatersleben, to the laboratory led by Ingo Schubert, and there I started to study chromosome biology, and that was my first touch with plant, let's say, epigenetics and chromatin. So afterwards I went to Ottron Mittelsteinscheid's lab at the Gregor Mendel Institute in Vienna, Austria, and there I really fell in love with epigenetics and chromatin, because I was studying effects of heat stress on plant epigenome, and that really like influenced my research direction for the future. And I continued, I combined this epigenetic research and chromatin research with studies of genome stability throughout my independent career as a group leader at the Max Planck. And I continue in this research direction until now, and after working for a very long time on Arabidopsis, I also added barley research to it, which is having much bigger genome, much more heterochromatin, and this creates some unique situation, new challenges, and also this genome is working under a little bit different principles.
Very nice. I find fascinating that you started being a hunter and in the end it's chromatin in plants. Like, I find that many people go to plants because they don't want to kill animals. So me included.
Me too.
Yes.
Yeah, it was initial idea, but at the end, it turned into, like, plants are very fascinating
Yes.
organisms, very unique, working on in like being rooted in the ground, and the top part is performing completely different function, like assimilates, is in contact with the light, so they are really fascinating organisms. So
For sure.
I'm happy to work with plants.
Yes.
Yes.
I think many of us share the same passion. So now we are going to the second part. Now we would like to enter a new session and discuss the EpiSeedLink Consortium. The EpiSeedLink Consortium is a project funded by the European Union's Horizon Europe Research and Innovation Programme under the Marie Slowodska-Curie grant. We dive into different topics such as seed germination, development, abiotic stress, and seed priming in 2 different species of interest, the famous and easy to grow model plant with the simple genome Arabidopsis thaliana and the crop brassica names, which give us a rapeseed oil. In your case, why did you decide to join EpiSeedLink? How does our research align with your own?
So I'm the member of the EpiSeedLink advisory board. So I did not decide to join myself, but I was kindly invited by the leader of the EpiSeedLink, Vicente Rubio, and it was really, I accepted this offer to join EpiSeedLink as advisory board member immediately. I did not need to think about it, because it's a great opportunity to see like lots of interesting research done by colleagues which are really great scientists, and it's also a great chance for me to learn communication in science and to be in contact with a very interesting scientific topic and with the next generation of scientists, basically. So what do I have in common with EpiSeedLink? And I think it's the team of epigenetics, because I work on epigenetics over the last 15 years, and also the seed phenotype, seed development, because with multiple Arabidopsis DNA repair mutants, we saw deformations or problems in seed development. So we looked on seed developmental phenotypes multiple times, and now with barley research we focus on genomic imprinting, which is connected with endosperm and also seed development. So I think I have a lot with EpiSeedLink, and it's really great to see how EpiSeedLink studies like early stages of plant, let's say, germination, and how this could be improved for the purpose of plant agriculture. So I'm learning a lot at EpiSeedLink.
Well, I am very happy that you have joined us in EpiSeedLink. Actually, today you gave a very nice talk about all the projects going on in your lab, and I think that just like matched perfectly what we were seeing for our projects in EpiSeedLink. And I also want to add that Aleš is a part of my thesis advisory committee, and I always look forward to present to him what I have done in my research in the past few months, because he always has very, very good advice. So going on to the next question. So I wanted to say now that we're in the last year of our consortium, and after 2.5 years, some of our fellows are in the last year of their PhDs for those who with 3 years of PhD, and others are entering the second half for those with 4 years of PhD, which is my case, and I think also Mayra's.
Yes.
And I wanted to ask you, Alice, what do you think about the research being done by EpiSeedLink?
So I think that there is lots of great research done at EpiSeedLink. I see steep progress also since the last meeting we had. So there was like in the first two meetings, because I think that's now the third time we are meeting. In the first two meetings, it was more like developing the ideas. The fellows were getting familiar with their projects. They were learning lots of methods. But now at this meeting, I see steep progress in terms of like data generation. So now many projects, they got their shape, and now the project ideas are very clear, and you are just following the main line of the project and getting data which will end up I guess very soon in many projects in like very nice scientific publications, which means you accumulated enough knowledge, enough new data to put it together and to communicate it to the scientific community. So what I think is really like lots of great stories coming up in EpiSeedLink, and I see also that many fellows, practically all fellows, really made great progress in their projects and in kind of like reaching scientific maturity, you know, like becoming real scientists, like fully established scientists and personalities, you know. So it's really great to see this progress.
Thanks. I must say that in the first meeting that we had, it was like 1 month after we
Yes.
arrived, we were very lost. So we were learning about our projects. The second meeting, we were more like, okay, we understand now, we are getting there. And this meeting, I think that we know what we are doing, and we are very excited to show you next year
Definitely.
our results. We are now entering the session regarding career. The beginning and development of a scientific career is a long marathon, with many uncertainties and challenges. What advice would you give a PhD student who aspires to have a fruitful career in research such as yours?
So I think a very important thing is you need to fall in love with your project. So if you really like your project, if you know you like what you are doing, then you can also handle some uncomfortable situation. You can get easier over hard times, which are associated with almost every project. There are very few projects which just run smoothly from the beginning till the end. Very often we as scientists have to solve lots of problems of technical character, sometimes generating materials, you know, establishing new methods which are not existing in the lab. So for all this, if you believe in your project, if you think it makes sense, that's gonna help a lot. I think this is one of the main things which scientists need to have at the beginning, to have this kind of like passion for this project, and if you fall in love with it, then this can bring you a long way.
Thank you, Alice. I think that is indeed very important. And as Mayra mentioned, a scientific career is a marathon, but there are sometimes very special or scary, actually, moments that lead us up to the next step. And I wanted to ask you if you have encountered pivoting points in your career, and if so, how would you advise a young researcher to handle those special or scary moments?
I think one one advice I want to give is you should realize that you are not alone in science. There are always people around which are helpful. Almost everybody in science has kind of like is willing to help, is a good colleague, you know. So talk to people you trust at your level, to the PhD fellows, talk to postdocs you trust, and finally talk also to your PI or to PIs you trust, because they are going to help you. So if you encounter a situation which is difficult for you, talk to people, because usually problems in science repeat. So what you are experiencing and is very new for you, other people in the lab might have experienced as well, because we all had to complete our PhD, we had to complete our first postdoc, we were all looking for positions at some point. All of us saw these grant rejections, paper rejections, and so on. So I mean, we had to handle these situations, and we might have already some experience which we are happy to share with you. So I would say there are very few situations in science which at the end cannot be handled in kind of like good way. So usually there is good solutions. Sometimes it requires more patience. Sometimes it requires like changing experimental design. Sometimes it requires rewriting your manuscript. But by talking to people, you can very often find like a reasonable solution to your problems.
Thanks a lot for your advice. I think that I have experienced this a lot in EpiSeedLink, because we are a group of fellows, and sometimes we are in the WhatsApp group just like, so this didn't work, help someone. And also like my PI especially likes to network a lot, so she just find a problem just like, okay, we are going to ask this person. So I really see the good thing of having people around you that can work you and work and help you.
I think the biggest lesson I've learned so far is to ask for help.
Yes.
Yeah. Just a lot of help. You will need it.
I think you as a student, you know, in this kind of digital time, you have really advantage of being connected, having your community. So the EpiSeedLink already makes the first network in your scientific career, and you might benefit from this network for very, very long time, you know, because the fellows, the PIs, you know, now they are all over the Europe, and you really have like a rich environment, rich network of contacts, which of people which are really great in science, great mentors, great colleagues, and they're gonna help you, you know, many times in the future.
Yes. Thanks a lot. Unfortunately, this is the end of our interview. Alice, thank you very much for sparing some of your time to share with us about your research, career, and your experience in the EpiSeedLink Consortium.
Yes, thank you very much, Alice. It was very nice to listen to some of your experiences and your very valuable advice. And I wanted to ask if you have any final remarks that you would like to share with us.
Yeah, I would like to thank you for inviting me for this interview. Thank you, Nora. Thank you, Myra. It's really great to be part of EpiSeedLink. And, you know, as you are learning some things in science, I'm also learning a lot about communication, you know, like how such consortium works. And I think it's really great environment for both the fellows and also the PIs, you know, for all of us to kind of like be in contact, to learn different things, because I mean, we are all the time in science experiencing new things, we are developing new concepts, we are trying to make science kind of like a good environment for people, you know, of different opinions, you know, like with different ideas, and try to make the best basically out of this what the scientific environment offers to us. So it's really great time talking to you here, and I'm really looking forward to see all the data in EpiSeedLink and the publications at the end which will come. And I also wish to all fellows like all the best and bright future.
Thank you again, Alice, and thank you everyone for listening. I hope you enjoyed this episode. Bye!
See you. Bye-bye.
Bye. Thank you, Aleš. Thank you, everyone who's listening.
Thank you for listening, and check out the other episodes of our EpiSeedLink podcast.